Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
It's kind of crazy how viruses, even ones that are like closely related, can have such different effects on us.
Yeah.
You got dengue and West Nile. They cause some nasty illnesses for sure.
Yeah, they do.
But then you have Zika virus, which is kind of in that same family. And it does something totally different. It can actually pass from a pregnant woman to her developing baby.
Right.
And it often leads to some pretty serious birth defects. Like, how does Zika manage to do that?
Yeah, that is the big question. Like, what makes Zika so unique? And it's a question that a lot of researchers are really digging into.
Right.
And there's some really fascinating new research that's starting to shed some light on how Zika actually does this, how it pulls off this kind of dangerous feat.
So we're going to break that down today.
Yeah.
Kind of a deep dive into that. So what's the actual mechanism? How does Zika get past that placental barrier and cause all this trouble?
Well, it's not just about getting across, it's about how it establishes itself within the placenta, which is that vital connection between the mother and the developing baby. And even how it gets the resources it needs to thrive and multiply in there.
So today, we're kind of diving deep into how does it do this? And we've got some new research to unpack. What are we looking at today?
Well, primarily, we're looking at a scientific paper that just came out that looks specifically at how Zika virus interacts with the cells in the placenta.
Okay.
And then just to give us a little bit of broader context about why this research is so important, we're also going to touch on some information from a recent health news article.
Got it. So for those of you listening, today's goal is to kind of break down this complex science in a way that is hopefully clear and engaging.
Yeah.
And really highlight what's important. What do you really need to know? We want you to walk away from this with a deeper understanding of just how resourceful viruses can be.
Right.
And how researchers are working hard to try and understand their secrets so we can prevent, hopefully, some of those scary consequences. So let's start with the study itself. What were the researchers hoping to achieve with this research?
The main goal was to understand exactly how Zika virus is transmitted from a mother to her fetus during pregnancy. Like, what are the steps involved? And they really zeroed in on how the virus spreads within the placenta itself.
Okay.
Because, that's the connection between the mother's body and the developing baby.
Makes sense. So what was the most, surprising or maybe groundbreaking finding that came out of this research?
The most significant thing they found was that Zika virus actually causes the formation of these long, thin connections between placental cells, which are called tunneling nanotubes.
Tunneling nanotubes.
Yeah.
That's a mouthful.
It is. It is.
So those are literally little tunnels between cells.
Yeah, you can think of them like little bridges or tiny pipelines that form between neighboring cells. And they create these direct pathways for all kinds of stuff to move from one cell to the next. Think of it as a secret passageway for the virus.
Okay, so is this different than just normal cell communication?
It is, yeah. So cells have different ways of talking to each other and exchanging things.
Right.
But these tunneling nanotubes are special because they form these direct open connections. Yeah. It's not just sending signals back and forth.
Yeah.
It's a physical connection.
Cool.
They're also surprisingly long. I mean, we're talking up to 700 micrometers sometimes, which is a huge distance on a cellular level.
Oh.
And they're mostly made up of these proteins called actin and tubulin, which are like building blocks inside the cell.
Got it. So how did the researchers figure out that this was happening? You can't just look at a cell and see a nanotube, right?
No, they had to be pretty clever about it.
Yeah.
They actually created the special version of Zika virus that had a little fluorescent tag attached to it.
Okay.
So they could see it, basically, under a microscope. And then as they were watching these infected cells, they saw these long, thin structures forming, connecting the infected cells to their uninfected neighbors.
So they literally could see the virus moving through these tunnels.
Yeah, pretty much. That's a simplified way of putting it, but essentially, yes.
Okay, cool.
Yeah.
And you mentioned different placental cells?
Yeah.
So which cells were they looking at specifically?
They focused on three main types: First, they looked at extravillous trophoblasts, which are cells that help anchor the placenta to the uterus. And they used this particular line of those cells called HTR8.
Okay.
Then they looked at cytotrophoblasts, which are important cells in the placenta's outer layer. And for those, they used a cell line called GG3.
Okay.
And finally, they also studied primary human trophoblast cells. And those were actually obtained directly from placentas.
Okay, so pretty good representation of the different cells that make up the placenta.
Exactly.
And we talked about how Zika is kind of unique.
Yeah.
Compared to some of its relatives like dengue or West Nile. So did the researchers look at whether those viruses could also cause these nanotube things to form?
Yeah, they did. And that was a really key part of the study.
Okay.
They wanted to see if this was something that was unique to Zika. So they looked at other flaviruses, like you mentioned, dengue and yellow fever.
Yeah.
And they found that while Zika virus, both the African and American strains, could easily trigger those cells to form tunneling nanotubes.
Oh, okay.
Dengue and yellow fever didn't do it. So it really points to this ability to induce these cellular bridges.
So that's Zika's.
It's like a Zika-specific thing.
A little trick.
Yeah, exactly.
Okay.
Right.
So if these tunnels are forming, what's actually moving through them?
Well, the virus can basically use these nanotubes to transport all sorts of things to its neighboring cells.
Okay.
So we're talking like actual virus particles themselves.
Okay.
Viral genetic material, which is called RNA.
Got it.
And even specific viral proteins.
Okay, so it's like they're passing on the infection without like coming out of the cells and floating around.
Yeah, they don't have to release like fully formed virus into the surrounding environment. They can just pass it directly to their neighbors.
Got it. And I think the study looked even deeper
It did.
into like what was causing these things to form in the first place.
Yeah, they wanted to pinpoint the exact culprit.
Uh-huh.
And so through their research, they actually identified a specific protein made by Zika virus called NS1, or non-structural protein 1.
Okay.
And they found that this protein was single-handedly responsible for kickstarting the formation of these tunnels.
So this protein's like the construction worker.
Yeah, exactly. It's like Zika's secret weapon for building these little secret passageways.
Okay. Is that like unusual for a protein, for a non-structural protein, I guess, to have that role?
Well, non-structural proteins are known to do a lot of different things, like messing with the cells they infect, for example. But this particular role of Zika's NS1 in causing these long-range connections, it seems to be pretty unique.
Okay.
And to make it even more interesting, they looked at NS1 proteins from other flaviviruses, like dengue, West Nile, and Japanese encephalitis. And they found that those didn't have the same effect.
Oh.
Like West Nile's NS1 could make a few tunnels, but nothing compared to Zika's.
Okay, so it's not just any NS1, it's Zika's particular.
It's got to be Zika's special NS1.
Okay.
Yeah.
And did they figure out like which part of that protein was responsible for that?
They did. They did even more experiments. And they figured out that it's the very first 50 amino acids at the beginning of the protein, which is called the N-terminus. That's the crucial part. And they actually created this mutant version of Zika that couldn't form those connections by messing with that region. They called it the Zika virus delta TNT mutant.
Zika virus delta TNT mutant. Got it. So they basically just took out the tunnel building part of the virus, and then what happened?
Well, that's where things get really interesting.
Okay.
Because they found that this mutant virus, the one that couldn't form the nanotubes, actually triggered a much stronger immune response in the cells than the regular Zika virus.
Interesting. So what do you mean by immune response? Like the cells were fighting back harder?
Exactly. So specifically, they saw an increase in the production of interferon lambda, which is like this signaling molecule that helps the body fight off viruses. And the regular Zika virus, the one that could make the tunnels, didn't cause as much of a reaction.
So it's almost like those tunnels are allowing Zika to sneak in under the radar.
Exactly. It's like a stealth mode. Because by using those direct connections, Zika doesn't have to release as much virus into the surrounding space. Which is what would normally trigger a stronger immune response.
So it's kind of hiding out a little bit.
It's hiding. It's being sneaky.
Okay, that's pretty cool. Now, the study also touched on how Zika gets the energy it needs to replicate.
Yes, that's another really interesting aspect of this research.
It was talking about taking resources from the cell or something?
Yeah, they found that Zika's NS1 protein interacts with a bunch of proteins that are found in mitochondria.
Oh.
And mitochondria are the powerhouses of the cell.
Right.
They're responsible for producing energy. So they noticed that cells that were infected with Zika, or even just producing the NS1 protein, they had more mitochondria than usual.
Interesting. Where are these extra mitochondria coming from?
Well, that's the cool part. They actually saw, using live cell imaging, that cells making Zika's NS1 could actually steal mitochondria.
What?
Yeah. They could take them from neighboring healthy cells.
Steal their power.
Yeah, basically.
Oh, wow.
You guessed it, through those tunneling nanotubes.
Way.
Yeah. Those tunnels are multitasking.
Wow.
And they found that the mutant NS1, the one that couldn't form the tunnels, was also bad at stealing mitochondria.
So it's not just spreading the virus, it's also stealing power.
Exactly.
It's like a double whammy.
It's rigging the system.
That's crazy. And is that mitochondrial theft really important for the virus?
It seems to be.
Yeah.
Because the researchers found that if they messed with the function or movement of mitochondria, like by using certain drugs, it actually made it harder for Zika to replicate.
Okay.
So it suggests that the virus needs those healthy mitochondria, whether it steals them or makes its own,
Got it.
to get the energy it needs to replicate.
It's like it needs that power boost.
Yeah, it's got to fuel its spread.
Okay, that makes sense. And you mentioned earlier that different placental cells might be more or less susceptible to Zika.
Yeah.
Did they find any differences in how well they made those tunnels or how they responded to the virus?
Yes, they did. It turns out that different types of placental cells, like those HTR8 cells and the JEG3 cells,
Right.
they actually had different abilities to form those tunneling nanotubes when they were infected with Zika. And they also had varying abilities to mount an immune response.
Oh, okay.
So that could be why Zika seems to infect different parts of the placenta
Okay.
with different levels of effectiveness.
Interesting. So some cells might be better at making the tunnels and therefore better at spreading the virus, I guess.
Exactly. Some cells might be more welcoming to the virus, so to speak.
Okay. So just to kind of summarize, this study showed that Zika has this unique ability to build these cellular tunnels using its NS1 protein.
Yeah.
And these tunnels then serve multiple purposes. They let the virus spread really easily from one cell to another. And it kind of looks like it helps it avoid the immune system. And then it's also using those same tunnels to steal mitochondria from healthy cells.
Yeah, it's got this whole system worked out.
Which gives it that energy boost.
It's pretty impressive in a scary kind of way.
Yeah. And you mentioned that this helps explain how Zika gets from a mother to her baby.
Right. So this research really shows us a new way that Zika is operating within the placenta. And by creating this sort of protected network, it can move between cells very stealthily.
Okay.
And it's kind of shielded from those main immune defenses. So it makes sense that this would play a role in its ability to be transmitted from mother to child.
And it's not just that it's getting in, it's like it's setting up shop.
Yeah.
It's taking what it needs.
It's making itself comfortable.
So that's pretty scary. But on the flip side, does this new understanding of how Zika works, does that give us any potential leads for treatment or prevention?
Oh, absolutely. I think this is where it gets really exciting, because now that we know that NS1, and specifically that N-terminal region, is so important for both the tunnel formation and the mitochondrial hijacking, it opens up a whole new world of possibilities for therapeutic targets. So we could potentially develop drugs or vaccines that could specifically interfere with those processes.
So block the virus from building those tunnels or from stealing mitochondria.
Exactly. And if we can do that, we could really limit its spread and hopefully prevent those devastating effects during pregnancy.
That's really promising. And, we talk about how it affects developing babies. Does understanding how it infects the placenta also give us more insight into why it causes those developmental problems?
Definitely. Because now we're starting to understand how the virus establishes itself and how it spreads within the placenta, which is, that critical connection for fetal development. And that gives us a better idea of how it might be crossing into the fetal circulation and affecting those developing tissues, especially the brain.
Got it. And the fact that this tunnel thing seems to be pretty unique to Zika.
Yeah.
It really highlights the need for treatments specifically designed for Zika.
Exactly. You can't just assume that what works for dengue or West Nile is going to work for Zika. Because it's got its own special way of doing things.
It's got its own tricks.
Okay.
So any research has limitations, of course.
Yeah, that's a really important point.
What are some things to keep in mind with this one?
Well, the first thing is that most of the study was done using cell cultures in a lab,
Okay.
which is a great way to get some initial insights.
Right.
But it doesn't perfectly replicate what happens in a real human placenta,
Okay.
A real placenta is much more complex.
Right. It's not just those 3 cell types.
Exactly. It's a whole ecosystem in there.
Okay, so it's possible that what they're seeing in the lab might not translate perfectly to a real pregnancy?
Yeah, that's always a possibility. And another thing is that while they know that the N-terminal part of NS1 is important,
Okay.
they don't know exactly how it works on a molecular level. Like, what are all the steps involved in making those tunnels?
So there's still some stuff to figure out there.
Absolutely.
Okay.
Yeah.
And was there anything else?
Oh, yeah. They mainly focused on interferon lambda,
Okay.
when they were looking at the immune response. But there are a lot of other ways that the immune system can fight off viruses.
Right.
And they didn't really look into whether the mutant virus might be affecting those other parts of the immune system differently, too.
So there could be more to the story.
Yeah, viruses are tricky.
Okay. So the next step, I guess, would be to test these findings out in a more complex setting.
Right, like in an animal model.
Right.
To see how it all works in a living organism.
Got it. So stepping back for a second, what are some of the bigger takeaways from all of this?
Right.
What do you think is the most important thing that we learned here?
Well, I think the most fascinating thing is how Zika has evolved this really specific tool.
NS1.
Yeah, the NS1.
Okay.
To do all these things, not only to help it spread directly between cells,
Right.
but also to manipulate the resources of those cells and even to kind of dampen down the immune response.
Like it's got a whole strategy.
It does. It's very strategic.
Mhm.
And I think it really highlights just how adaptable viruses are.
Right.
They're constantly evolving and finding new ways to survive.
And does this make you think about other viruses that might be using similar tactics?
Oh, absolutely.
Like other ones that can cross the placenta?
Yeah, because there are other viruses that can do that.
Right.
And now we're wondering, are they using similar tricks?
Yeah. And I guess we don't know that yet.
Not yet, but it's definitely something to look into.
Yeah, that's really interesting. So for anyone listening, as you're thinking about this, this whole back and forth between a virus and our cells,
Yeah.
think about what this means. If we can figure out these strategies that viruses are using,
Yeah.
what other stealth mechanisms are out there?
Right.
And how can we use that knowledge to protect ourselves and future generations?
It's a constant arms race.
Yeah. So if you're interested in learning more,
Yeah.
you could look into the research that's being done on other viruses that can affect pregnancy,
Yeah.
or research into new antiviral therapies that are being developed.
That's a great suggestion. And there's a lot of information out there for anyone who wants to dig deeper.
Awesome.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.